Assalamualaikum warahmatullahi wabarakatuh. Bismillahirrahmanirrahim. My name is Asma, and I am here to share and present on the topic of my paper review, which is overview of cellulase, enhancing of cellulase productions, and transgenic cellulolytic plant. Before that, we went through the introductions, which is the purpose. On the purpose of the paper review is to review on the cellulose productions by microorganisms and transgenic plant. And basically, the cellulose enzymes consist of three types of enzyme, which is endoglucanase, beta-glucosidase, and azoglucanase. And these three types of enzyme, which are normally and commonly work as synergistically, which is functions together, while at several times they functions individually as well. And the microorganisms basically produce have the potentials in productions of cellulase. Why? Because the microorganisms have this high stability in the cellulase productions in order to hydrolyze cellulose into glucose. And the sources of microorganisms are many types in environments, which is you can have your microorganisms from soil, from animals, from plant, and even from the biomass. And the applications of the cellulase also have become in various of industries like textile industries, pulp and papers industries, as well as biofuels industries, and also in food processing and food productions like bakery and juices or beverage industries. For the in methodology, which is I have read on the 70 papers, including 10 of 70 is review papers, and the balance is 60 journals, 60 journal articles, and whereby only 50 papers of journal articles that have been cited in that I have cited in my paper review. And the sources of the journals basically from the international journals, and only a few from the local journal, as well as the sources of the journals I have browsed and searched on the engines of Scopus, Science Direct, Research Case, and also Springer links. And first and foremost, it is the contents of my the main content basically in my paper review, and which is on the identifications of cellulase producing microorganisms or bacteria. And there are three few types of enzyme assay that can be used in the identifications, where the normally have been done on the DNS method, which is a 3,4-dinitrosillic method, which is on the identifications of either beta-glucosidase or endoglucanase, or even the azoglucanase identifications, and also filter paper, ammonium sulfate precipitations, and also Mendel's Racy mediums. The identifications of cellulase depend on the microorganisms species, and also the carbon sources of the during the purifications and extractions of the cellulase. And basically, for example, the Aspergillus niger, which is has been done on the carbon source as from the Archis hypochaea shell, which is the extractions of the enzymes produced is basically endoglucanase, by using the NS method as enzyme assay, while compared to Bacillus mortis with RG07, which is with carbon sources from the carboxymethyl cellulase, it can produce endoglucanase, azoglucanase, and beta-glucosidase, which is the carbon sources is carboxyl methyl cellulose, and the enzyme assay used is ammonium sulfate precipitations. And this is the part of another part of the content in my paper review, which is on the inducer that can be produce high amounts of cellulase producing bacteria, which is there are a few techniques, which is genetic manipulations, hydrolysis substances, fusion transcription factor, and also gene knockout. And for the genetic manipulations, basically multi-gene expression manipulations have been done on the Trichodermariaceae, which is the Trichodermariaceae is the fungus, filamentous fungus that have had famous in the cellulose productions. And also for the hydrolysis substances, bioconversion element has been used as a hydrolysis substances like as paper, biogas, waste from environments like plant waste, food waste, and many more. And next is fusion transcription factor is used to release the carbon catabolite repressions, which is CCR, that have in gene inhibitor in cellulose productions. Therefore, when we are releasing the carbon catabolite repressions, the cellulose productions can be increased immediately. And then the gene knockout is which is the analyzing on the unknown gene functions initially, and after that we have done on the deletions. They had done on the deleting the genes inhibition in the cellulose productions. Therefore, after you use these techniques of inducing the for inducing the cellular productions, and the cellulose productions can be increased in the yield. Transgenic plant expressing cellulolytic enzyme, which is the technique to have been done on the which is cellulolytic enzyme, has been implanted or has been engineered inside of the plant cells, therefore to increase their stability or even the cellulose functions in the plant. For example, genetic engineered maize which with cellulase, which is the main identifications is to determine the stability of genetic inside of the maize, and also to understand the transgenic hybrid of the characteristic of the maize when they are grown and planted. And for the second is transgenic of rice and tobacco. And the transgenic of rice and tobacco basically is to during the cellulose to increase the high yield of sugar during the sacrifications of cellulose of the rice and tobacco, and whereby the high yield of sugar can be used as for many functions in the industries, like food industries, biofuels, and many more. Last but not least is on the rice transgenic plant, which is the other types of identifications, which is they had identified that cell wall of the plant of rice transgenic, which is have the positions of the cell wall, and also have reduced in the crystalline of cellulose, whereby the consequences is the cellulose hydrolysis can be reduced in times, and the productions of the saccharifications process will be done immediately without consuming a long time. I think this is the conclusions of my part in the paper review, which is applications of cellulose in the industries are widely distributed. Therefore, the cellulose engineered have been produced to produce the cellulose in a bulk quantity for applications in many industries. And transgenic plant, I believe, have the opportunity to be in future applications to reduce biomass abundance in the environment as well. And the recommendations here is to further investigate in the transgenic plant expressing cellulase, especially as the source of the journal informations in this age is limited in the limited informations that I have found, and cellulase producing bacteria identifications can be improved in their structure and also empowering the technique of screening of cellulase, and I believe that it is believed that all the various sources of microorganisms are available in the future. And this is the references that have been put in their presentations during the presentations. And I think that's all from my part in the presenting my paper review. Thank you for your attentions, and Assalamualaikum warahmatullahi wabarakatuh.